Swayam Prabha. Digital India. Educated India.
Welcome to the first module of Plant Biotechnology course. This is the first lecture in this module, and in this module we will talk about plant tissue culture in general, where we would discuss variety of topics including totipotancy, differentiation, somatic embryogenesis, various parameters that impact the in vitro regeneration, micropropagation, initiation of cultures from diverse type of explants, and so on. All these topics will be covered in this unit. My name is Manoj Sharma and I am working as an assistant professor of biology at Jawaharlal Nehru University, New Delhi. In JNU, I teach plant biotechnology and genetic engineering at School of Biotechnology. This project is sponsored by DTH Swayamprabha, MHRD, New Delhi. So in today's lecture, we will talk about mainly cellular totipotancy, organogenesis, and somatic embryogenesis. Cellular totipotancy. So as per the standard definition of cellular totipotancy or the totipotancy, it is the genetic potential or the capability of the differentiated or specialized cells to regenerate into a complete plant. This is called as cellular totipotancy. In other words, it can be said as it is a characteristic of plant cells where the potential of forming all the tissues or organs or organ types is retained with the cells even when these cells mature to differentiate into specialized forms to perform specialized functions. So therefore, theoretically we can say as long as a plant cell is alive, that it has a intact membrane system and it has a viable nucleus, it is capable of reverting back to meristematic state, and hence the complete plants can be produced from these cells. Now, as the cells divide and gain specialized forms, animal cells usually loses the potential to grow into a new organism. However, in plant cells, all the living plants, all the cells retain this capability to revert back to the meristematic state on exposure to the favorable environmental conditions. So it's very important that though capability is there, but yes, those induction conditions are required which will induce these cells to regenerate into a complete plants. So therefore, we can use any plant tissue or organ like a stem, leaf, or a root segment for the initiation of culture. So like this is a tomato seedling, and we can use any of its living part, like we can use the leaf segments from here, or we can use the small stem segment. We can cut the small stem segment and we can use them as a explant to start the culture, or even we can use the root segment or the root apical meristem to initiate the culture. And these plant parts or the tissues or the cells which are used to initiate these cultures they are called as the explants. Even a single cell from a suspension culture or can be used to initiate this process of regeneration, and full protein plant can be formed. So in the culture, they usually in a standard they are de-differentiated to form a undifferentiated mass of cells, which is called callus, and then that callus is induced for the morphogenic responses, which result into the production of these complete plants. Therefore, all the living plant cells, irrespective to their specialization status or the physiological status, has the potential to revert back to the meristematic state when these are exposed to favorable cultural conditions, and they can regenerate into a complete plant. Now, before going ahead, let's learn about few important terms that we will be using frequently in the following lectures. The first is the differentiation. A process by which a cell acquires modification in its form and function is called as differentiation. In plants, meristematic cells, they divide and give rise to different type of cells which form later tissues and organs and have the specialized function with the specialized forms. In fact, in the multicellular organisms, including animal and plants, the whole body is actually derived from a single cell, that is the zygote, which is formed by the fusion of the male and female gametes. The zygote divide and redivide, and through the process of differentiation, it specializes to form the special tissues and organs, and which results into the formation of a complete body. Now, the cells which that undergo this differentiation and gain a specialized forms to perform special functions are called as differentiated cells. Second is dedifferentiation. So dedifferentiation is literally the opposite of differentiation. So it is the process or the phenomena, cellular process, in which the mature or differentiated cells lose their special forms, lose their special status, and revert back to the meristematic state is called as dedifferentiation. In the differentiation, less specialized cells are transformed into more specialized cells. In dedifferentiation, more specialized cells are revert back to the less specialized forms which perform the generalized functions, and the cells obtained through the process of dedifferentiation, they are called as dedifferentiated cells. Next is redifferentiation. So when dedifferentiated cells are induced for the morphogenetic responses or are induced for the differentiation, this process is called as a redifferentiation. So it's basically simulates exactly the differentiation. However, the starting material here are the dedifferentiated cells, and hence it is called as a redifferentiation. Another is transdifferentiation. The process by which a cell type present in one tissue or organ converts to a specialized cell type or of other tissue or other organ without going through the process of dedifferentiation. That is called as a transdifferentiation. So in other words, transdifferentiation refers to the conversion of a differentiated cell of one particular developmental commitment to a differentiated cell of another developmental commitment. Like here is the leaf segment. It has a mesophyll cells or the leaf cells, different kind of leaf cells, which are filled with the chlorophyll and chloroplast, and the main function of these cells is the photosynthesis. And when they are induced to dedifferentiate, they make callus. And this process of induction or when the specialized cells are losing the status to become the undifferentiated cell, this is called as a dedifferentiation. And the Callus cells then can be induced to form any specialized kind of cells. It may be different kind of shooting responses, maybe the rooting response, or any particular specific type of cells can be initiated or induced from these undifferentiated cells. Like here, Trachary cells are induced. Trachary cells are the part of the xylem which are involved in the transport of solutes in plants, and this process is called as a redifferentiation. However, if these cells of this particular developmental commitment, if they transform directly to the tissue type of a different development commitment from the photosynthesis to the transport without the process of dedifferentiation or redifferentiation, then this process is called as transdifferentiation. Another term is regeneration. So typically the generation of entire plant from a cultured explant is called as regeneration. Regeneration may occur by a callus formation or directly from the explant, as we saw for the transdifferentiation, where we saw that how the one cell type is directly changing into another specialized cell type. And another most commonly used term would be a callus. So the callus is a mass of proliferating, unorganized or undifferentiated cells which is produced from any plant part, tissue or organ that are separated from the parent plant and are grown aseptically on the nutrient media under controlled environmental conditions. Usually callus are grown on the solid substrate where all the media or nutritional components are provided in the solid substrates. So let's discuss now about the organogenesis. So the standard definition of organogenesis is it's a process of differentiation which involves the formation of various organs like shoots, roots, etc. from the vegetative plant tissues or from the cultured plant cells or from the undifferentiated mass of cells, that is the callus. So actually this is just the differentiation as we discussed in the earlier slides. However, here it is restricted to the formation of organs only, and hence it is called as the organogenesis. Now, as we discussed earlier, any of the explant, any of the plant part can be used for the production of these in vitro cultures to initiate the in vitro culture. However, these explants, which are plant parts which are used, they should be mitotically active cells. Hence, the regions on the plant which has the mitotically active tissue, usually explant should be prepared from those regions, because the distribution or the physiological status of the different tissues or different organs in the plant body is varies from one tissue to another. Hence, the tissues which are mitotically active and physiologically younger or physiologically active, they are usually used to initiate the cultures. So theoretically, again, the tissue should be mitotically active, as we discussed earlier. Now, this is kind of organogenesis is one of very important aspects of the plant tissue culture and is widely used for in vitro regeneration in plant tissue culture. So plant growth regulators or other media component, nutritional components, they play a specific role or significant roles in this process of organogenesis. So it's a very important commercial aspect. Usually organogenesis may take place directly from the cells. They may direct without the formation of callus, but more common is indirect, where we go through the callus formation and then dedifferentiation. So here is the list of various explants which are usually used in one or other species, like apical meristems. Most of these tissues which are highlighted here or marked here are physiologically active and mitotically active, and hence they can be used, like the apical meristems. Continuously dividing cells are there. Leaves, so specifically here we are talking about the younger leaves, younger growing leaves, not the mature leaves. Axillary meristems, or the stems which are the younger growing stems, or the cotyledons, hypocotyls, roots, or root to apical meristems. So again, we are talking about the younger tissues or the mitotically active tissues. In addition to this, once the plant matures, fluorescence grows, so the fluorescence parts, younger fluorescence parts, or even the immature embryos from the developing seeds are also used for initiating the in vitro cultures. Now, different species may respond very differently to these different kind of explants. Like the physiological status of a leaf in a young seedling, young dicot seedling versus a young monocot seedling is very different. And the regeneration potential, it has been found that the leaves from the dicot can be used, or the younger leaves from the dicots can be used to initiate the cultures. However, leaf tissue as an explant is not successful or very successful for initiating the in vitro cultures in the monocots. So the actual success of these meristem or these explants would depend on the physiological status and mitotic activity status of these cells, or the cells present in those explants, and may differ from one species to another species significantly. One may be successful, leaf may be successful in one species and it may not be successful in other species, like we just discussed about monocot and dicots. The organogenesis may be grouped or put into the two categories depending upon which pathway does it use for the production or for the regeneration of the organs. One is the direct organogenesis, that is the development of the new organs directly from the explants. And second is the indirect organogenesis, that is the development of the new organs through the callus formation. As we saw earlier also, the difference between the transdifferentiation and redifferentiation. So in the indirect organogenesis, the explants which are more specialized, usually they are dedifferentiated into less specialized forms or undifferentiated cells, that is the callus, which are then evoked for the morphogenetic responses through the supply of growth regulators or the media components, and organs are regenerated, whereas direct organogenesis is the production of organs without the formation of callus. So the direct organogenesis, when we transfer any these organs or tissues on a medium supplemented with the growth hormones, these somatic tissues, they are capable of regenerating into adventitious buds or the shoots, or the initiate the new shoots or the new terminal meristems, and these buds are formed directly from the plant organ. There is no callus production, no dedifferentiation, and any piece of tissue can result in the formation of these kind of structures. And this type of organogenesis is called as the direct organogenesis. So here is the example. What we see is an apical meristem is, or the shoot apical meristem is excised, and then it is induced to form the multiple shoot tips or the multiple buds at the same region. Now they are induced to proliferate, they grow further, and then they are separated from each other and transferred onto the rooting media individually. Now, when they are separated and placed individually, once they produce these root buds or the roots, they grow into the media. Each of the seedling becomes, each of the plantlet or the shoot becomes an independent plantlet, and then they can be transferred onto the soil for the acclimation, and new complete plants, separate plants are regenerated. So this type of organogenesis, where callus formation or dedifferentiation is not involved, is called as a direct organogenesis. Second is the indirect organogenesis. So indirect organogenesis is the development of the new organ through the callus formation. In general, in plant tissue culture, indirect organogenesis is more common as compared to the direct organogenesis. Like here is the leaf tissue, which is specialized. The cells of leaves are specialized to form the photosynthetic functions, photosynthesis related functions, and now when they are induced for the dedifferentiation in response to the growth regulators, these cells form the callus, undifferentiated mass of these mitotically active cells, and then these are induced to evoke the morphogenetic responses like the shooting response and then the rooting response, which leads to the production of new plantlets, and this is a redifferentiation. So this process where specialized cells are dedifferentiated onto the callus, and then the callus or undifferentiated cells are induced to form the organs, is called as a indirect organogenesis. Typically, any of the explant can be used for indirect organogenesis. The indirect organogenesis potential of different organs of on the plant body is also higher as compared to the direct organogenesis. Direct organogenesis, in fact, is hypothesized or explained based upon the predetermination theory. That means the cells need to have a predetermination or pre-induction for the production of these organs. Only then they can be initiated or induced by providing the artificial culture environment or nutritional environment. So indirect organogenesis is more common and more successful in tissue cultures. So technically, any of the explants can be used to initiate the culture, and here is an example of cotyledon explants from the tomato, which are harvested, and they are small segments are made from the cotyledons and which are being used for the in vitro culture. So they are incubated on the callus induction media, and this induces the formation of this mass of dedifferentiated cells, or the mass of callus. So basically this process here, the first step, the callus induction, is actually the dedifferentiation of the specialized cells to the less specialized cells to perform the generalized functions. And once the enough amount of the callus has been formed, and then they are transferred onto the shoot induction media. So now we won't need to supplement the media components or the growth regulators which are required to initiate the morphogenic responses from the from this calli or undifferentiated mass of cells. Now on incubation onto the shoot induction media, in few days this callus regenerate or develop the shoot buds, which later forms the shoot or small shoot apical meristem, which grow into the small shoots. Once the shoots are allowed to proliferate, so that sufficient when they grow in a sufficient size, like maybe 4 to 6 centimeter or 7 centimeter, we need to transfer them onto the rooting media. So it depends upon the culture vessel. If we are growing these calli, or if we are inducing the shoot induction in the Petri plates, so for the proliferation or elongation of the shoots, we need to transfer these calli into the large jam bottles. So once we transfer once the shoots sufficient length of the shoots is achieved, sufficient elongation has occurred, we transfer them onto the rooting media where root induction takes place. Once these shoots, they produce the roots, they becomes the complete plantlets, and now these plantlets are ready to transfer onto the soil, and they start behave independent plants. So basically here, through the using the cotyledonary explants, we harvested the cotyledonary explants from the tomato seedlings. We induced the dedifferentiation process, where specialized cells were transformed into less specialized cells or undifferentiated cells, which later now be evoked using the growth regulators or the other nutritional media components. We evoked the morphogenic responses or a differentiation, cytodifferentiation responses, which led to the development of the shooting response from this media. We let the shoot grow, elongate. Once the sufficient length is achieved, we transfer them onto the rooting media, and finally our plantlets will be ready for transplantation onto the soil. And in this way, any of the explants can be used for redifferentiation and this process of organogenesis. Now we will talk about somatic embryogenesis. So embryogenesis in general represents the development period when a zygote undergo a series of differentiation events leading to the formation of mature embryo. This differentiation usually involves a series of predetermined pattern of cell division, organ formation, which lead to the formation of a mature embryo. So if we in the plants or in plants or the animal, this zygotic or the embryogenesis, it takes place. It starts with the fusion of the male or the female gamete that produces the zygote. So this zygote now divide and redivide in a manner or in a series of predetermined patterns of the cell division, and lead to the formation of more specialized tissues, leading to the finally formation of the fully mature embryo. Now this embryo in the plant possesses two distinct regions, the mature embryo. This is the plumule and radicle. So the one leads to the production or the formation of the shoot apical meristem of the shoots, and the second lead to the production of or the formation of roots when the embryo germinates. And then we have these cotyledonary leaves, which the first leaves appear when it germinates and appear from the seed. So embryogenesis, this whole process of embryogenesis, where male and female gametes are involved, and the fertilization of male and gamete is involved to formation of the zygotes and leading to the formation of embryo. This whole process is restricted to the specialized reproductive structures, and like the ovules or the embryo sac, and the fertilization is essential for the production of zygotic embryos. Now fertilization is essential for the production of zygotic embryos, but fertilization independent embryogenesis do exist in nature too. Now what exactly that means? That means that the somatic cells, any of the somatic cells or the sporophytic cells from the any part of the plant may be able to make the somatic embryos. It does exist in nature, however, in nature it occurs in the sporogenic tissue or sporophytic tissue, which is associated with the reproductive cells. So what exactly happens is, like, this is the somatic cells or the sporogenous tissue in the reproductive cells, and it is induced, it gets the induction signal, and these cells, some of these cells, they gain the induction to differentiate or initiate the process of somatic embryogenesis, leading to the formation of full-fledged complete embryos as we saw in the zygotic embryos. Like the zygotic embryos, this asexual or the somatic embryos also have well differentiated two distinct regions, one which lead to the production of the shoots and and the roots, and these embryos which are produced from the somatic cells, these are called as asexual embryos or the somatic embryos. In some plants like the citrus and mangoes, these adventive or asexual embryos are naturally formed from the nucellus tissue. Now, this phenomena of development of the embryo from somatic tissues or the callus tissue is called as somatic embryogenesis. It may be initiated or induced from even the single cells in the suspension culture, and embryos produced as a result of this somatic embryogenesis are called as somatic embryos or asexual embryos. So now, because of this process, as for the zygotic embryos, the fertilization of the male and female gamete was essential. However, this zygotic embryos, which which can immediately germinate into a complete plant, or they may later be allowed to germinate into a complete plant. This adds to our understanding that probably the fertilization process or the fertilization of the male and female gamete is not absolutely essential for the embryogenesis or the development of the embryos in plants, and so it is something related to the specific environment in the reproductive cells or the specialized structures like ovule or the embryo sac, where which result in the induction of these embryos. So if we can replicate the environment in these reproductive structures artificially or in vitro, we can induce the somatic cells to form the somatic embryos, which can lead to the formation of later germinate into the complete plant. So over the period of last 3 or 4 decades, these factors has been well understood. The role of various nutrients, role of various growth regulators or the vitamins, how they affect the cultures. We will discuss later in today's lecture too. So it has been made possible to initiate the somatic embryogenesis in the in vitro culture. Now, depending upon what pathway does it take, it can be grouped or put into the two categories, either direct somatic embryogenesis or indirect somatic embryogenesis. So like the organogenesis, same phenomena, same pathways are here. Development of the new somatic embryos, that is directly from the explant tissue without the production of callus. And in the indirect, embryogenesis process, the specialized tissue first are de-differentiated to form the undifferentiated mass of cells, and later they are induced to form the develop the embryos from these callus cells. So That is a indirect or a direct somatic embryogenesis. Let's have a look on the somatic or the direct somatic embryogenesis. So, like these are the somatic cells, these somatic cells are transferred onto this tissue is transferred onto the induction media or the induction component in the form of growth regulators and other nutritional component. They are provided, and it leads to uh, to the the initiation or induction of it may be in a single cell or a group of cells that start developing the embryonic response and ultimately lead to the formation of full-fledged embryos which are completely ready to germinate. They may immediately they can immediately germinate and can form the full-fledged plant. So the isolated cells in the suspension culture or the protoplast of alpha have been successfully induced for the somatic embryogenesis. So it's not only that actually the organs are required to induce the somatic embryogenesis. Even the isolated cells, single cells, or even the protoplast, where protoplasts are the cells or when the cell wall is removed, what is the remaining part of the cell, that is the protoplast. So even the protoplast can be induced to form the somatic embryos, and it has been successfully achieved in this system, that is the alpha plant system. So in other words, so now this is direct embryogenesis. It's not that common in nature. So in other words, it is defined, or it is hypothesized or theorized based on the inherent capacity of the embryo production. So it states that in order to initiate the direct somatic embryos from the somatic cells need to have an inherent capacity of embryo production, and this is facilitated by the optimal artificial culture condition or the culture environment that lead to the induction of these somatic embryos followed by the production of the somatic embryos. Later at some time we will also learn that how these somatic embryos can be used for the production of various applications like the synthetic seeds. Indirect somatic embryogenesis, as again it's like it's the same phenomena or hypothesis as it was for indirect organogenesis, that is the formation of undifferentiated mass of cells or the callus from the explant followed by the development of embryos from these cells. So explant cells are usually specialized, we know about it. Like any of the explants we can use, maybe the apical meristem or the leaf or the cotyledon or the stem cuttings. All these cells, they have a specialized status, they are performing the specialized functions. So the first step of the indirect somatic embryogenesis is the dedifferentiation. We induce the dedifferentiation in these somatic cells, and it results into this dedifferentiation step result into the formation of callus structure, or that is the mass of undifferentiated cells which are mitotically active. Now once the enough mass of cells is accumulated, we transfer them onto the somatic embryogenic media or the embryogenesis media, or the media which is required, or the regulators or the nutritional components that is required to induce the somatic embryogenesis. They are provided, and during this time what happens is that some of the cells, they gain the potential, embryogenic potential, and they start developing into the embryos. These embryos, once their cells are determined to produce the embryos, they grow and form the full-fledged embryos, which are again have the distinct region of apical shoot development and the root development and cotyledon type of structure, which are not actually cotyledon, but those kind of structures are there. and then finally, when they germinate, they are they make the complete plants. So the artificial conditions for the induction of the somatic embryos are well optimized in several systems. However, carrot is one of the systems which is really well worked out, and hence carrot cells are a good model system to study the various aspects of somatic embryogenesis. Now, the in vitro cultures, whether the organogenesis or the somatic embryogenesis or the redifferentiation or the differentiation or the redifferentiation, these all in vitro culture conditions, they are directly regulated by several parameters which include or which can be grouped in three broad categories. We have biological factors, media or nutritional components, and physical factors. So the biological factors includes various parameters which are associated with the starting material, that is the explant, and these parameters can directly regulate the response of these tissues on the culture environment. Like the explant source, what was the physiological status of the cells in this in the tissue from where they were harvested, whether these explants were harvested from highly differentiated cells or whether the tissue was mitotically dormant or physiologically dormant or more or less highly specialized tissue. So depending upon that, there will be a direct impact. Obviously, the highest specialized cells, they are difficult to de-differentiate, or more optimization would be required for de-differentiating these explants or these cells. Similarly, the age of the explants, with age the explants mature, plant cells gain maturity, and so the younger the tissue, better the response of these tissues to the culture media would be. So the physiological status, from where it is harvested, in which condition plant is growing, whether the plant is stressed or not, how healthy the plant is, all these factors which are associated with the source of the source plant, they can have a huge impact. Now, second is the media composition or nutritional components. Now, the selection or the optimization of the right culture media has a huge impact on the success of any plant tissue culture environment. These media components, they play huge role, and it's a very tedious, or it is very important to optimize the correct combination, to identify the correct combination for culturing requirement for one particular species, and it's specifically hard for the recalcitrant species. So various media salts which include inorganic or the organic components, or inorganic salts like different elements, inorganic elements, then the organic source like the carbon source or the vitamins or the amino acids are supplied sometime. So all these media components, and then very important, that is the phytohormones. So these are the phytohormones or the growth regulators. They have a huge impact on these on the in vitro culturing. So the growth hormones, they specifically they are required to evoke the morphogenetic responses in the plants, and generally they are required in very low concentration, very minute concentration. However, their impact is significant, and the requirement of the different species may be very different for the supply or the concentration or amount of these hormones. Also, the response of these hormones may differ at their different concentrations. Like if we use the auxins at a very low concentration, it may evoke the rooting responses. However, if we increase the concentration of the auxins, it usually evokes the callusing responses, that is the dedifferentiation. So again, the different explants from the same species or explants from the different species, they behave differently. And even the different components or the different type of the auxins or different kind of the cytokinins, they have their different impact. Their impact may be very different. Similarly, the physical factors, which include pH of the media where we culture the explants, light requirements, temperature requirements in the culture room, humidity in the vessel, culture vessel, or the gaseous environment in the culture vessel can have a direct impact or interfere with the culturing conditions or the culturing responses of these explants to the in vitro cultures. So different species might have different light and temperature requirements, and so accordingly, depending upon the species, we need to optimize these parameters. For example, for the somatic embryogenesis in Solanum, it may require, or it do require, the presence of light. However, if we want to incubate or want to evoke the somatic embryogenic response in popular species, Calli from the popular species, we need to incubate them in the absolute darkness. So these are the species-specific responses, and we need to independently optimize them. And moreover, it is not only always the species-specific, even the different explant from the same species, same plant, or they may respond differently. Or even many times, the different conditions or the different steps of the organogenesis may need different incubations. For example, generally the optimum temperature for the tissue culture, in vitro culture, is usually 24 to 28 degrees Celsius. However, in some cases, in some species, incubation of the callus at the low temperature increases the efficiency of the shooting response. Like in the tobacco itself, the response of the shooting response at about 18 to 20 degrees Celsius is much better as compared to when for the shooting these calli are incubated around at 24 or 20 in the range of 24 to 28 degrees Celsius. So we will certainly discuss more about these media components and composition in the plant tissue culture media, and a little bit about biological and physical factors also in more detail. What should be keep in mind while initiating these cultures in our next lecture. So that's it for today. Quickly, if we talk about what we discussed today, overall, we discussed about the cellular totipotancy, and then we discussed about the organogenesis, how it is being established, what kind of organogenesis is there, and then same for the somatic embryogenesis, its establishment and the different pathways of somatic embryogenesis. So that's it for today's this lecture. Thank you.